Alrighty, it is 9 o'clock, so we will go ahead and get started. Thank you everyone for participating in this month's GVN webinar, and it is my pleasure to introduce our speaker, Dr. Helena Solo-Gabriele. She is a professor in the Department of Chemical, Environmental, and Materials Engineering at the University of Miami. As a faculty member, she teaches courses in environmental measurements, water and wastewater treatment plant design, and environmental engineering microbiology. Her research focuses on problems that relate the environment to human health. During the COVID-19 pandemic, her research team used wastewater to track illnesses within campus dormitories, the university hospital, and within Miami-Dade County Public Schools, and for the entire county through a major centralized wastewater treatment plant. Since the pandemic, her team has expanded measurements to include additional respiratory viruses, including Influenza A and B, RSV, in addition to poliovirus, Mpox, and C. auris. Most recently, her team has initiated studies to utilize wastewater to track dengue virus within Miami-Dade County and Florida. During this presentation, she's going to describe the results, emphasizing the utility of wastewater in tracking disease, and will indicate next steps for tracking dengue and other arboviruses. So thank you, everyone, and please join me in welcoming Dr. Solo-Gabriele.
Thank you so much. I'm going to go ahead and share my screen. And do you see my screen? Yes?
Yes.
Thank you. And thank you so much for that introduction. I really appreciate it. And I appreciate everyone's time in listening to our work on wastewater-based epidemiology. As mentioned, I'll be talking about using wastewater to track disease prevalence. This is a project that stems from a consortium that we put together called SF for South Florida, Radical. And our funding came predominantly from the NIH during the height of the pandemic, through the RADx Radical program, and then also through RADx Up. Up stands for underprivileged communities. And we also receive supplemental support through 4Catalyzer. This project is a joint project between University of Miami and Weill Cornell Medicine. It was a 3 PI project, including myself, Helena Solo-Gabriele, professor, as mentioned, and my collaborator, Stefan Schur, also from the University of Miami. He's a data scientist. And Chris Mason, who is from Weill Cornell Medicine, and he's a bioinformatician. Our aims are listed here. Specifically, our goal was to develop a data standardization platform that can take information from human surveillance or clinical data, combine it with wastewater data, put it together so that we can predict and provide recommendations about what's going on in terms of the prevalence of disease within communities and about its transmission. Our hope in providing this data to our decision makers is that they could use it to develop policies. So with that, I'd like to go ahead and continue. But before I start, I wanted to emphasize that this project is a huge team effort. Many people were involved. And I especially wanted to acknowledge the University of Miami, the upper leadership of the University of Miami, President Frank, who is a public health expert. Our university was the only university that remained open during the pandemic, mainly because of very aggressive public health surveillance program that was set up for our university. Included the collaboration of many units of the university, including our community member, Miami-Dade Water and Sewer, and of course, Weill Cornell Medicine. Again, just to emphasize, during the COVID pandemic, it was all hands on deck. All units of the university came together to develop and implement a public health program to maintain the university open while maintaining everyone safe. It included collaboration through the Sylvester Comprehensive Cancer Center, the University of Miami Hospital, the University of Miami Center for AIDS Research, the University Miller School of Medicine, the College of Engineering, of course, Weill Cornell Medicine, and Metasub through Weill Cornell Medicine. So the background of our study is that we always knew that when people were sick, especially with gastrointestinal disease, that they would release their organisms, infectious agents into the sewer. But it wasn't until the COVID-19 pandemic that we really realized that it's not only limited to gastrointestinal disease, but respiratory diseases can also be tracked, and even skin diseases can be tracked through the sewer system. We release body fluids, all kinds of body fluids, and then all of this ends up in the sewer. So we have a huge repository of community disease information within the sewer system. And so our goal was to capitalize on this, to collect samples from the sewer system and then purify it and analyze it for specific disease targets. During the COVID-19 pandemic, it was COVID-19. It was SARS-CoV-2. So our objective is to relate what we see in the wastewater to predict disease prevalence within a community. So that was our ultimate goal, is to relate the wastewater to disease. Our workflows for most of we did several projects, and this is the general workflow that we use for the majority of our projects, which involve first collecting the sample, either a grab sample or a composite sample from the sewer system. That sample then underwent a series of splits. One of the main splits was through electronegative filtration, where we would acidify the sample and capture the viruses on the filters. We would prepare 3 filters. One filter for Mark Sharkey's lab at the Center for AIDS Research, another filter for the Oncogenomics Shared Resource, which is managed by Sean Williams, and then also a third filter that went to the Weill Cornell Medicine to Chris Mason's lab. Once these filters were prepared, they underwent different processes. The first set of filters was used by Mark Sharkey. He developed a new technology called Volcano Second Generation, or V2G qPCR, which is a fast and rapid technology, less expensive than the more traditional RT-qPCR. And that's what we used on a day-to-day basis. We would collect the samples in the morning, and then by the afternoon, we had the data ready and prepared reports and would issue them within a period of 1 day. And so the V2G allowed us to do that. Hand in hand, we were comparing our results with the RT-qPCR. And then also through the Oncogenomics Shared Resource, the yellow, our team conducted targeted sequencing. So sequencing focused on identifying the variants of SARS-CoV-2 as those different variants were coming through the communities. At Weill Cornell Medicine, those filters were used predominantly for bulk or shotgun sequencing, RNA-seq. Also, all the sequencing data went to Weill Cornell Medicine, both the targeted sequencing from the Oncogenomics Shared Resource and also the bulk sequencing that was conducted through Weill Cornell. Our design of our sampling programs were always paired with a human surveillance program. As I mentioned before, the University of Miami had a very aggressive tracking process for our students, for our faculty, and our staff. And so we had incredible amount of information and data on a building by building basis within our university. We knew the number of residents within individual dorms that were positive, given the clinical results in any given week, for example. In addition to our information from our campus population, we also were able to get data from our university hospital. The university hospital has the advantage in that the number of COVID patients is very well documented. They know how many are positive within the hospital. Plus, we can gather information on comorbidities of those patients in the hospital. In addition to that, in Miami-Dade County, the Florida Department of Health was monitoring positives on a zip code basis, and they were aggregating the data on a day-by-day and zip code basis. And we also were able to get data through Miami-Dade County Public Schools from 9 schools in northern Miami-Dade County. At the schools, it was difficult to get information specifically on COVID positives through the families that were at the schools. So instead, at the schools, we use absenteeism as a proxy for illness at the schools. In this presentation, as I mentioned, we have done several different projects focused on wastewater-based epidemiology. I wanted to lump these projects into 3 areas of emphasis. The first area of emphasis focuses on illustrating and emphasizing the power of disease tracking that we observed during the COVID-19 pandemic. We observed during the pandemic that wastewater-based epidemiology was capable of predicting cases and hospitalizations. In addition to that, wastewater-based epidemiology was capable of identifying variants in the wastewater, and we were also able to establish relationships with comorbidities and other disease targets. After the height of the COVID-19 pandemic, we continued with the wastewater-based monitoring, looking at longer-term trends in wastewater-based monitoring, and also focused efforts on expanding to new targets. In this longer-term analysis, we established the importance of temporal, of how to separate the data in terms of variant waves, and also illustrate the importance of spatial aggregation in understanding trends within the community. In addition to that, we showed that for other disease targets that they show a lot of promise in terms of detecting percent positives within a community. I wanted to end the presentation with the third area of emphasis, which focuses on describing the advances in sequencing. The advances of sequencing include the use of bulk sequencing to identify disease targets instead of the qPCR, which is more targeted. And then also we have experimented with host attribution using a new way of sequencing that provides information about host attribution, which I'll describe at the end. So coming back to our sample collection plans where our wastewater samples were paired with information about human health, I just wanted to give you an example of what we saw in terms of the hospital. So on the vertical axis, we have the hospitalizations, and the black line corresponds to the hospitalizations within our community, within Miami-Dade County. And then on the right axis, we have wastewater, and the green corresponds to the wastewater numbers. And you can see that the wastewater shows an increase in levels prior to the increase in the hospitalizations. So the information from the wastewater was very effective at predicting hospitalizations in the future. And as a matter of fact, through our colleague, Bavar Shukla, he and colleagues were able to develop a model that in part used information from the wastewater to predict allocation needs or resource needs within the hospital. And as a result, the University of Miami Hospital was able to optimize the need for COVID-19 rooms versus other rooms that can be used for other types of procedures. So it's very valuable in terms of being able to allocate, not only tracking disease prevalence, but allocate resources within a hospital. Also, we see during the COVID pandemic, when the progression of the vaccines became available, the number of hospitalizations declined, but then we got various waves coming through associated with the variants, as illustrated here. We see that at the beginning, we had lower levels of SARS-CoV-2 in wastewater relative towards later times. We had higher levels of SARS-CoV-2. However, the hospitalizations were higher at the beginning and lower at the end. So we also note that although the wastewater tracks the trends of hospitalizations, the one-to-one relationship varied, where at the beginning, a little bit of SARS-CoV-2 in the wastewater meant a lot of hospitalizations later on, whereas at the end, towards the latter parts of the pandemic, a lot of SARS-CoV-2 in the wastewater did not translate to the same number of hospitalizations. That had to do with the intensity of the disease as it waned with the various variants. So essentially, if we looked at the relationship between wastewater and hospitalization from the initial wave to the Delta wave, to the Omicron, to the post-Omicron wave, the flattening of the curve, where the relationship became flatter and flatter between what we saw in the wastewater and what we saw in terms of hospitalizations. In terms of the variants, on the right-hand side, we have the data on the variants showing the different colors show the different variants. And on the top, we have the data from the clinical laboratory, the Oncogenomics Shared Resource, analyzed clinical samples, as illustrated here, but also analyzed the wastewater samples shown on the bottom. And we see that there's pretty much a correspondence. You can see a correspondence between the different colors as the pandemic proceeded. In addition to that, what we know, there's a general correspondence, but if we look at each individual variant in more detail, we can see that in some cases, wastewater, we saw the variant in wastewater before it was observed in the clinical samples. So the wastewater not only tells us that there's going to be more cases of SARS of COVID-19, but it even tells us when the variants have hit our community. We knew that the Delta variant, we were aware of the Delta variant in the international arena. We knew it was going to come, and we were able to see it in the wastewater 7 days in the Miami-Dade community, 7 days prior to it being observed in the clinical samples, as an example. In terms of the hospital wastewater, again, the hospital wastewater was a great resource of information. Of course, with the hospital, the levels of SARS-CoV-2 in the wastewater correlated with the number of patients. And early during the pandemic, it correlated with remdesivir administration. Also, at the height of the COVID-19 pandemic, there was also a second outbreak of monkeypox. And during this period of time, we showed from the hospital wastewater when there were clinical cases of Mpox at the hospital during July and August, we also observed elevated levels of monkeypox within the wastewater. So this showed that not only can we track COVID-19, but we can track other viral targets. And not only can we identify viral targets, we can identify fungal targets, as Candida auris is illustrated here, where we have our clinical cases shown in blue. So during the pandemic, there were anywhere from 15 to 30 individuals in the hospital that were positive for Candida auris. There was a lot this is considered to be a lot of patients, mainly because during the pandemic, they didn't have a way of moving the patients out of the hospital if they were positive for Candida auris. It was common that people would come in with another ailment, another illness, but they were also positive for Candida auris. In the hospital, they would get better from their original illness, but they couldn't leave because there were no places that were willing at that time to accept the Candida auris positive patients. And so a lot of the patients just stayed in the hospital, and that's why we had the number of patients that we had. But what was very interesting was that in the wastewater, even though Candida auris is predominantly a skin ailment, we were able to see Candida auris in the wastewater intermittently when we had the clinical patients within the hospital positive for Candida auris. And these orange dots correspond to positives by qPCR. But what's really interesting is that we also cultured the samples and grew for Candida species, and we were able to identify Candida auris actually in the cultured samples. And so not only is the Candida auris nucleic acids found in the wastewater, but there's viable Candida within the wastewater capable of growing in culture. So that brings us to our second area of emphasis, which is on the long-term measurements and expansion to new targets. Here we were able to expand to additional collaborators, including Yale University, through the funding that we received from Catalyzer. And the questions that we wanted to ask with the long-term data is illustrated or stated here. First is we wanted to see how what was the relationship between SARS-CoV-2 and human clinical data, and how did they compare across adjacent areas and over time. Also, we wanted to explore question number 2, which was to look at other viral targets. Can they be adequately surveyed through wastewater? The clinical data that we had available to us included COVID PCR tests, the number of tests by day and by zip code. And we also had the number of positives by day and by zip code. And we had this data available for a period of 46 months, almost 4 years. In terms of the wastewater data, we had 5 laboratories. We had data from 5 laboratories. 2 of the laboratories were at University of Miami, the Center for AIDS Research, and the Oncogenomics Shared Resource. Of course, we sent samples over to Yale, and then there were also, the wastewater treatment plant also had access to additional data through Wastewater Scan and BioBot. All of these laboratories, they all ran different things, but all of them were consistent in running SARS-CoV-2. So that's the data that I will illustrate here. But before I talk about the actual data, I want to explain how the data is organized. And in terms of the human health data, the data is organized. We have the data available by zip code. But our wastewater treatment plants, we have 3 wastewater treatment plants in Miami-Dade County. We have the North District that serves this part of the county up here. We have the Central District, which serves the central part of the county. And then we have the South District, which serves the southern part of the county. So since the wastewater plant is in these larger areas, we had to aggregate the health data by region to coincide with the area serviced by the wastewater treatment plant. In addition to that, through our data that was available through the Department of Health, we had positive cases and we had test counts. Because we had test counts, we can then therefore calculate the positivity rate, which is the ratio of positive cases to test counts. Also, the human health data was available on a daily basis, but our wastewater data was more or less pretty much on a weekly basis. So we aggregated the human health data on a weekly basis. Also, given the variability between week to week and the analysis themselves, we then established moving averages. We then took the weekly data and averaged it over a 3-week period to get a 3-week moving average for the human data. The wastewater data, again, it was in weekly basis. We computed moving average, 3-week moving averages, and then to establish the overall combined impacts of concentrations of targets, we did a weighted average based on the number of people that are serviced by each of these wastewater plants, given the values of target relative to the population. So we did a weighted average to account for an overall combined based on population differences. To illustrate the data, on the left-hand side, we have the positive case counts, and the black bars correspond to the positive PCR cases over time. And what you'll see is, of course, we have the different waves coming through. This one is the Omicron wave. And then it starts to decline. The PCR positive cases start declining. And it's mainly because people are not getting tested as much as they were at the height of the pandemic. But if we superimpose positivity, which is the ratio of positives to the number of tests, then we can look at the positivity, and we see that as the pandemic proceeded over time, the positivity, you can see the waves very clearly coming through. So amongst those that were being tested, there was still a high percentage that were testing positive. So the positivity is more sensitive and can take into account the fact that testing numbers went down over time. We compare that to the wastewater on the right-hand axis, superimpose the wastewater data. And for the most part, the wastewater shows the trends, mimics the trends more or less during the Delta wave, during the Omicron wave, during the post-Omicron wave, and the subsequent post-Omicron waves shown there. So coming back to our first question about whether or not there's a correlation between the cases and the wastewater numbers. We know that there were higher, we look at the three wastewater treatment plant data within Miami-Dade County. Remember, there were 3, north, central, and south. If we focus our attention to the pink here, which is the central plant, the population that were serviced by the central plant had the highest testing numbers. And then those, because they had such high testing numbers, they also had the highest positive case counts. Taking the ratio of the two to establish positivity, then the positivity, however, within the central area tend to be low. Yes, and I think we're going to put somebody on mute. Elizabeth or Ruby, thank you. Okay, so the positivity rate was lowest at the central plant, even though they had the highest number of PCR, and it's because the testing was so high. But for the most part, what we see is that all the peaks coincide with one another at each one of the plants. And if we put all of the plant data together, again, it's consistent with what we see for the individual plants, where we have the positivity, again, showing in the red, dominating. So to address our question number 1 about correlations, what we know is that the raw data here is shown on the top. The green and the black correspond to, the black is the number of PCR positives. The green is the number of tests The red is the positivity, so that's the health data, but then the wastewater is superimposed on it through the brown line for one lab and the blue line for another lab. And if we compare the correlations between the different categories, we have positivity, positive cases, and testing. And green means better correlation. So what we're looking for is areas that have more green. And if you, in looking at this plot or at this graph, we see much more green under positivity. So the positivity is providing us with better correlation with the human clinical data between positivity and wastewater. So we have a better correlation between the wastewater if we use positivity rates. Looking at the temporal scales of waves or variant waves, single year and grouped year, we see more green under the waves. So if we aggregate the data looking by waves, by variant waves, we get better correlations. And then if we start looking at which wastewater treatment plant, we see that the south plant has more green than the central and north plant. And we attribute the higher correlation of the south plant due to the geographic situation of the south area. So if we look at the south plant, Miami-Dade County is on the southeast peninsula, southeast tip of the Florida peninsula. And the South District plant is down even further, and we have the Atlantic Ocean on the east, and to the south we have the Florida Keys, which has a small population. There's much more movement of people across the northern boundary of Miami-Dade County, and we attribute the better correlation in the South District due to the more limited mobility of people across the county lines. Coming to question number 2, which focused on other targets. This is just an example of some of the other targets we looked at. This is PMMOV, which is peppered mild mottled virus, which is a dietary indicator. We eat peppers and then we release, and the peppers have the virus, and then those viruses get released through the feces. And so we use it to track human waste. And what we see is the PMMOV, when we start comparing across labs, the comparisons are not quite as robust as with SARS-CoV-2. But regardless, the trends are more or less can track one another, but the means are different. So the different labs for PMMOV provide different absolute numbers or means. So the means are different between different labs. Looking at the disease targets, such as influenza A, we compare, for example, lab 4 and 5 and lab 2. For example, lab 2 has very low positives, whereas 4 and 5 have very similar numbers of positives, although the positives don't necessarily coincide with one another. So the correlations are not that good, but the percent detects on positives between lab 4 and 5 was promising. Lab 2 was did not have as sensitive of a detection method. So we attribute the coincident, this variability at the low end due to the fact that we're near the detection limit for influenza within wastewater. And we believe that with further advancements and increase in sensitivity of the technology, that we will be able to be farther above the detection limit so that we can start seeing more correspondence. But we believe that this shows promise, and at least the percent detected was not different for 2 of the 3 laboratories. And we interpret this again as showing promise. There is promise in potentially having multiple laboratories run for additional viral disease targets. So that brings us to our third area of emphasis, which is the advances in the sequencing. And here in this third area, we added an additional collaborator, Phase Genomics. I wanted to first talk about bulk sequencing and the advances of bulk sequencing. Prior to the pandemic, environmental engineering microbiologists would use sequencing, but it was more to look at the microbial community at the phylum level, order level, class level. At that time, before the pandemic, it was not common to see levels that you can even see pathogens. The idea was, can we see the pathogens? And prior to the pandemic, it wasn't really, it was thought of to be in the future. But the future is here, And for example, this illustration shows that bulk sequencing, we're now able to see pathogens. In this particular image, we can see the norovirus and the Aichi virus, two potential pathogens in wastewater through bulk sequencing, not targeted, bulk sequencing. Similarly, for the bacteria, on the right-hand side, species-level bacteria are being observed within the wastewater now. So the pathogenic species level, which is pretty amazing. So now we can start looking at the wastewater without even just looking at the whole community, not targeting for a specific organism. You don't have to necessarily know what's there to see it, which is a new paradigm in monitoring wastewater. In addition to pathogens, we can also see antimicrobial resistance genes within the wastewater. What we see here is large diversity and number of antimicrobial resistance genes in hospital wastewater. What was also very interesting is that as we proceeded from the dormitory building scale to the campus basin down to the wastewater treatment plant, the number and diversity of the AMR genes decreased, which was an interesting observation that we weren't aware of before. But this brings us to host attribution. The host attribution is especially powerful for identifying the host for AMR genes in wastewater. The way this is done is through a special type of sequencing called high-C sequencing, high throughput chromosome conformation capture. The way the method works is through a process called proximity ligation, which associates the AMR gene to its host. The proximity ligation is done by using formaldehyde that binds different parts of DNA together, and then those are cut and then sequenced. And so that process provides us with information about the gene location. So that AMR gene, we now know the gene location. Is it located in the host genome? Is it located within a plasmid? Or is it in a bacteriophage? In addition to that, it provides information as to the host, who is carrying it, which bacteria is carrying it. And it provides data at the species level, genus and species level, which is pretty amazing. So now we can associate three things together. We can identify the AMR gene, we can identify which microbial host, which bacteria is carrying that AMR gene, and where is that AMR gene within the host? Is it in the plasmid? Is it in the genome? As an example. This is, you can't read this, but this is an example of the data that comes from the host attribution associations. This is output from phase genomics. On the horizontal axis are the different AMR genes. On the vertical axis are different bacteria. So as an example, we look at one bacteria. This one that's highlighted here is of the genus Prevotella. And we see two blue marks in there. That means that there are two AMR genes that were identified with the Prevotella. And that Prevotella was within the genome of the that AMR gene was within the genome of the Prevotella. So pretty amazing information that you can gather from the host attribution. Looking at the data a little differently, so we identified all the species that we could see with this host attribution. Again, this is a proof of concept. And for Bacteroides, gremlin-solvans, we're able to see beta-lactam within the beta-lactam AMR resistance within the plasmid. In addition to that, within the same bacteria, we're able to see resistance to tetracycline, to macrolide, and lincosamide. And that resistance is found within the core genome of the bacteria. So the amount of information and the associations that you get within the organism is pretty amazing with the high-C sequencing. So in conclusion, I just wanted to emphasize that wastewater-based epidemiology is a new transformative discipline that sort of exploded during the COVID-19 pandemic. It's a new approach, completely different, that augments the clinically based approaches that we've been using to track disease historically. Again, it was proven during the COVID-19 pandemic, and we know that it can not only track SARS-CoV-2. It can track other viral targets. It can track microbes from other domains, the bacteria, the fungi, protozoa. We also envision a transition of wastewater-based epidemiology, which currently focus a lot on qPCR, towards more and more sequencing, targeted and bulk sequencing. What we found through bulk and shotgun sequencing is that we are able to identify infectious microbes. That's the big advantage. You can identify the microbes even before you know they're there. So you don't necessarily need to know that there's SARS-CoV-2 or COVID-19 circulating in the community. Theoretically, you could look at the wastewater first to see what's there without knowing what is circulating. In addition to that, the sequencing has the added ability of doing host attribution studies. So overall, we see that there's a huge potential for wastewater-based epidemiology, um, huge potential for monitoring viruses globally, which is one of the objectives of the global virus networks. So with that, I'd like to thank everyone for listening, for your time. We have a QR code that can be used to access our webpage. I also want to mention that our papers, I have the last slide of our presentation has the list of our papers, and the DOIs are there in case you are interested in more details of our studies. And with that, I'd like to open it up for questions. Thank you.
Thank you so much. Incredible presentation. So for questions, we have the Q&A. Please type up your question and then I can read it aloud. And to start us off, our first question, since RNA can be degraded so rapidly, is it still possible to detect using these methods?
That was like the revelation that we got during COVID-19 in the environmental microbiology community, my community. We always thought that RNA was so fragile and would degrade so quickly. There was something about SARS-CoV-2. We don't know what it is, but it persists for a long time in wastewater. So there's some viruses that can persist for a long period of time. Now, whether or not other viruses do is another question. So it may be specific to the type of virus.
Thank you. So our next question, very insightful presentation. I was wondering how Candida auris eventually could influence the detectability of SARS-CoV-2 and variants of other viral targets in wastewater.
How Candida auris could impact SARS-CoV-2? We think that during the pandemic, there was, we did observe a lot of Candida, but it had more to do with the inability of people to move, and so it was concentrated in certain places, the Candida auris, because they weren't able to accept people in nursing homes, for example, with the long-term illness. So that's why we saw it in the wastewater. And we saw an association between the SARS-CoV-2 and the Candida auris in the hospital, for example.
Thank you. So great talk. Have you looked at evaluating for NTM such as M. abscessus or for M. leprae? Is it feasible?
So the mycobacterium, I believe it can definitely be seen. We did not analyze specifically for the mycobacterium, but we did provide wastewater samples to people who were measuring mycobacterium in the wastewater. There was one researcher who was looking at at phages for mycobacterium from the wastewater. I'm pretty sure we can see it. And there was a master student actually who did this work looking at mycobacterium and correlating it with illness in a community and in wastewater. So I have a vague memory of that. It can be observed in wastewater.
It's very exciting to be able to see that as well. Moving over to arboviruses, we have thanks for the great talk. Can you comment on wastewater as a possible tool for arbovirus surveillance?
That's what we're currently working on. We are currently working on using wastewater to monitor arboviruses. And a lot of, for example, here in Miami-Dade County, we're very vulnerable to the arboviruses because of our climate, a lot of mosquitoes, and then also because of our travel associated uh, imported cases. And so the surveillance for arboviruses right now rely a lot on people being diagnosed, but not everybody gets diagnosed within Miami-Dade County. And also monitoring the mosquito population. So we have been gathering samples for dengue analysis, and we're in the process of analyzing those samples to see if we can identify it. There's been another study conducted by Wastewater Scan Group, Wolf et al., that did find dengue the two serotypes of dengue within our wastewater here in Miami-Dade County. So we're pretty confident that it can be done, and we're in the process of developing those technologies.
Next question. Thank you for the presentation. I have a technical question. To improve representativeness, particularly in untargeted sequencing approaches, what increase in sequencing depth is generally recommended, and how do you determine the optimal coverage?
The sequencing is not my expertise, but I do listen to a lot of conversations of the experts. And they're always talking about 30 million as like the base and going down to 100 million and even multiples of 100 millions. So that's what I hear from my colleagues to that level.
Was any correlation analysis made between AMR genes and noroviruses in terms of host attribution to eventually look at how these two intersect?
We did not do that analysis or correlation to identify whether or not the AMR was related to norovirus, which is it's a good, we should do that. We have the data, we should go ahead and do it. And I think it's a good idea. We were more focused on the understanding the host attribution, which focuses predominantly, the data is predominantly on bacteria, looking at where the AMR lies within bacteria. But there may be a correlation with viruses that we're not aware of, but it would be worth looking at. Thank you.
Another question on host attribution. So for host attribution, is it possible to differentiate between human host, animal host, or if it is environmentally using sequencing?
When it came to wastewater, I know that we were the first step of running the wastewater samples so that we would not be defy IRB or human subjects is all human reads had to be removed. So from a sequencing perspective, I'm pretty sure that the technology exists to identify the human reads versus reads from other organisms. So it's done. It can be done. It is done.
So next question, excellent presentation. What, in your view, will be the major obstacles to reaching comprehensive surveillance throughout Florida?
Mm-hmm. Well, I think we just need to get buy-in from all agencies involved, from all the public health. And I do believe that there is. It's just a matter of getting organized and getting a framework by which all the data can be shared through the health departments in Florida. We have three big laboratories that have capacity, government laboratories that have capacity in Jacksonville, Tampa, and in Miami. It would be a matter of getting organized and developing a system to receive samples and a dashboard to post the data, automatic processing of the data to post the information on a dashboard, and then come up with a public, it's one thing to have a dashboard, but then you have to have a communications plan so that the community knows better, can make better informed decisions based on the information that's presented. So I think the technology is there. I think we can do it. It's a matter of getting organized.
Certainly, and that's almost always the hardest part.
Mhm.
Next question. Thank you for this presentation. It was very interesting. Is there a known reason why Delta had an increased hospitalization rate over wastewater detection as compared to other variants? Was it due to its high transmissibility, environmental conditions, or other factors?
I don't know for sure, but it's my understanding that the Delta variant was very intense and that people who were not everybody was vaccinated at that time. There was a transition. I mean, there was a lot of people the vaccine was available, but not everybody was vaccinated. And it was amongst those who were not vaccinated that really suffered the intensity of the illness. So I believe it had to do also with the vaccination rates as well. Um, I and it's also that from the pandemic, the transmissivity tended to increase as we went along. So Delta was more transmissive than the earlier variants, and then Omicron was even more transmissive than Delta. But the intensity of the disease also waned as time went on. But by the time Omicron came around, it was like 6 months later, more people had been vaccinated as well.
So next question, congrats on your presentation. I have a technical question as well. Has your group worked with microbial amplicon preparation within the wastewater samples?
The amplicon preparation was done through the oncogenomic shared resource where they amplified for SARS-CoV-2 so that they can look at the variants.
Okay.
So yes, from that perspective, yes. And then in traditional environmental microbiology, it's very common to amplify for the bacterial signals.
Thank you. And next question, great talk. Based on your experience, are there any suitable or better alternatives to pepper mild model virus? Would you recommend the use of multiple controls as an indicator of fecal contamination?
Hey, yeah, the pepper mild model virus is used a lot of laboratories use it, but there are other indicators of fecal waste that can be used. One that was developed through our laboratory, or human waste, human indicator waste, was developed by Mark Sharkey, B2M. It's a housekeeping gene from humans. And if it's detected in the wastewater, we know that there's human fluids in the wastewater. And we looked at the use of PMMOV and also B2M, but there are other ones as well. And to normalize our signals, because wastewater comes from a lot of different sources in a building. It's not only the toilet or the sinks and showers, but there's hand washing, there's clothes washing, there's dishwashing. So there's sources that have different variable amounts of human waste coming in. So you want an indicator that tells you the relative level of human waste so that you can normalize your sample by that indicator. So if you have a lot of waste, you'd expect a lot more of the target organism. But what we found is that there's variability in the waste indicator, and there's variability in the target. And we take you divide one by the other, there's more variability produced. So we found that when it came to these waste indicators, they were most useful when, for normalization, they were most useful for small watersheds, so for small scale. So if we're sampling at the building level, normalizing by the waste indicator was useful. But if we're doing a very large community, such as the entirety of Miami-Dade County, the waste indicator didn't really give us much, normalization by that waste indicator didn't give us that much more improvement in our correlations. So it's a function of scale. And then to answer the question about other indicators, other waste indicators, yes, there are a lot of them actually. It's not only pepper, they have a tomato rugose indicator, crassphage, in addition to the B2N. They can all be used to indicate waste. You always want to make sure that there's human fluids in there, regardless, even if you don't use it as a normalization factor.
Thank you. And our final question, so going back to the idea of a dashboard or some way to communicate data, what would your community engagement be like in regards to this discussion?
Again, our public health professionals, those who work with the community in public health would be the people that I think would be the ones who could best design a dashboard that would be useful and helpful for the community, so that they are able to make the best decisions possible. So for example, in terms of the children, when we were looking at the Miami-Dade County Public Schools, there was a lot of controversy about whether or not masks should be used and, what protections the family should take for their children. And, under those conditions, it would be useful for the families to be aware of when the spikes are happening within those individual schools. And at the school level, you can clearly see spikes coming through. It's very like on off kind of thing. And in a situation where you have a lot of people coming together, and I think it's best to have some level of information about, what is potentially circulating. Is it a respiratory disease? Is it a diarrheal disease? So that way people themselves can take precaution. But exactly how to convey that information, convert these numbers of genomic copies into something that people can easily assimilate. I think that's something that, the professionals in the public health field can do. There was some talk about color coding, green, yellow, red for, median, high alert levels versus low alert levels, that kind of thing. But they're people who have expertise in that would be the ones to develop that. Thank you.
Certainly. Thank you so much. So that is all of our questions. So once again, I would like to thank you for speaking today and sharing all of this incredible information on wastewater surveillance and wastewater-based epidemiology. I would like for everyone here to know about our next webinar, which is going to be held on March 24th at 10 a.m. with Dr. Fernando Spilkey. The title is going to be Virus Genomic Surveillance and One Health, diving into the Brazilian virosphere. Additionally, I would also like to point out that GVN has their annual International Scientific Meeting being held next week. You can find more information on our website. Thank you so much, everyone, and we look forward to seeing you at our next webinar.
Thank you.